But once we actually explain, no, wait, we're not crazy. Aging is a biological phenomenon, and there are species that live much longer than humans. So there's nothing fundamental about our 80-year time limit. So potentially it's just an engineering problem that we can at least increase it to something that is observed in other animals.
Welcome to Live Longer World, a podcast where we unite to boost longevity and fight aging. All resources and premium member benefits are found at livelongerworld.com. Now, on to today's episode. Hi, Yuri, and welcome to the Live Longer World podcast.
Thank you. It's a pleasure. Thanks for inviting me.
Yes, I'm very excited to talk to you. And I think particularly, Yuri, you're very outspoken and bold. Of course, it comes across in you uncovering the lab leak theory, but we'll first talk about longevity. We'll keep it to longevity. But you've spoken about, how you want 100% life extension. And I've heard you speak about, people sometimes in academia are okay with 10 to 20% life extension. And there's this missing sense of urgency in the field. You want 100% life extension, so do I. So why do you think there is this missing sense of urgency, or maybe what can be done to solve this problem?
I see you've been kind of doing your background research. Definitely hearing some of the things I've said in the past about the lack of sense of urgency in just kind of the academia of the aging research. And I think when I was kind of starting to say these things, we've made a lot of progress since then. I mean, because, probably like 5-6 years ago is when I was kind of complaining about this lack of sense of urgency, and since then we had a lot of investment come into the space, and I think it's generating a lot more urgency in the people who are being kind of hired by kind of people who want to see a much higher sense of urgency, and they're essentially giving them all the tools that they need to go as deep as they need into the science and generate first hypotheses, and hopefully they'll translate into therapies that will give us something. Well, if not 100% at this point, maybe 20-30% in a very short future. And especially like the 10-15% that we see in animal models of life extension that get a lot of play in academic circles, in conferences. I think for our purposes, of human life extension, they're nowhere near enough that we need to have even humans live 10-15% longer, because 10-15% in mice is really not translatable into 10-15% of humans. Like we have, I think my biggest kind of example of this is caloric restriction, which in mice can give you up to 50% life extension, but when they tried it in primates, in macaques, that only translated into like 5%, 10% lifespan increase, and it was gender or sex specific. So males didn't really get a lot of life extension, females did, but for that they needed to be like starved 2/3 of their lives, and so like you can see like 50% for mice get decreased into like just 5 to 10% in humans. So we really need to see big breakthroughs in animals, like large increases in lifespan, to be able to be confident that it'll translate into something meaningful in us.
And in fact, that starvation also, it comes with its own downsides, where you lose all your muscle and you become frail, and that's exactly the opposite of what you want.
Yeah, it's not a very enjoyable life. You might not want to extend it very much if you're actually like starving for most of your life.
Exactly. Yeah, no, it's quite exciting that there is so much more money that's come into the field, that people are becoming more aware of it. And then that point of awareness, you in fact did work in advocacy for aging for a long time, or just activism before you started your company. And you've written some amazing blog pieces as well on your Medium. So I'm curious, when you were involved more on the awareness side, are there certain explanations that you think worked in terms of convincing people or informing them more about this space? And on the flip side, maybe some explanations that just don't work and don't resonate with people?
Right. I think it just takes a lot of patience. Like this patience. That's what I learned, that, you can't convince people quickly. And the first time you're going to tell them something that, let's extend lifespan, they're probably going to recoil and say, let's not. They're going to say it's going to cause overpopulation, or, death gives meaning to life, or some other, excuse that we usually hear when people say they don't want to extend lifespan. So, but I think, if they actually are given an opportunity to kind of think about it, in privacy and without any kind of pressure on them to kind of stand their ground, they'll gradually come to the realization that, it's actually not a bad thing. There's nothing bad about being healthier for longer or living longer, as long as, you want to do it. So, and essentially, I think we have to raise awareness of that it's not just possible to extend lifespan, because many people just think, it's impossible. They kind of have this learned helplessness from a very early age, that aging is just some fundamental law of nature, and for some reason humans have to die at 80 years old, and that's kind of ingrained in our culture and everything else, and we kind of take it as a given. And when someone tells us otherwise, we tend to dismiss them as, someone crazy or something, because, it contradicts our beliefs. And but once we actually explain, no, wait, we're not crazy. Aging is a biological phenomenon, and there are species that live much longer than humans. So there's nothing fundamental about our 80-year, time limit. So potentially it's just an engineering problem that we can at least increase it to something that is observed in other animals, or even mammals. Whales live 200 years. We're mammals. There's probably nothing fundamental why we cannot live at least as long, or maybe even longer, like sharks, 400 years. So, and once I think you kind of plant the seeds, and people, rational people, will let those seeds grow into understanding a little bit that maybe the previous beliefs that we had culturally ingrained in ourselves are not really fundamental, and were based on kind of the previous facts on the ground, where at that point there was no genetic engineering, there was no biology, hundreds of thousands of years ago, and aging was an immutable law. But now it's not the same. Now the situation is different. So potentially we can do something about it, and we just have to very quickly understand what exactly makes us age in the way that we do and live as long as we do it, and change that biology. And that kind of ties back into the sense of urgency. We can take, a few decades to do that, and probably a lot of people won't be able to enjoy the fruits of that labor, or we can do it very quickly. And I think it's just a matter of how quickly the research can be done. And for that, I think we need a lot of people doing the research, and that kind of ties back into the funding. We need a lot of funding to fund these, a lot of people doing the research. And I think, ultimately this will help get us there. And once you kind of deliver this message to people, I think a lot of them will change their mind. And this is essentially, raising awareness that aging is not something untouchable. It's not something immutable. It's actually just a biological feature that we can probably change. Not probably, but we can change.
Yeah. Yeah, it's so interesting, right? When there's something that's so ingrained in human culture, we'll come up with all sorts of reasons to try to rationalize it in our head. And I think that flip too, when you think that something's uncontrollable, that you just accept it, and then you come up with different reasons where, it's religion, or it's leaving resources for the young, or what have you. But then once you have that switch and flip where it's like, wait, it can be controlled. Why should I die if I don't want to die? I should die if I want to die, not something that it's imposed on me from external factors.
Yeah, absolutely. And this is kind of what we're fighting against, is this our learned helplessness. I say a lot of times that we just kind of get raised with this thought that we have to die, and we have to accept that we're dying, and that we will die. And I think, kind of the first step is to rebel against this and say, well, I don't want to, and we should really do everything possible that we can to give people the opportunity at least to live longer, if not completely, avoid death.
Right. So how did you become aware of it? Because I know you have a background in mathematics, computer science by just formal education, and then you did work in pharma and drug discovery in Russia for a bit. How did you come into the field of longevity?
Right. Yeah, exactly. After I kind of transitioned from the IT and technical background into drug development, this is where I met a lot of people who are also into transhumanism, life extension, aging research in Russia. And I got kind of converted, or at least I was made aware. I went through all the same stages. You first hear about extending lifespan and you go, but what about overpopulation? And then you start thinking about it and then realize, yeah, there's nothing bad about extending lifespan, and just gotten kind of more and more interested about just the kind of the movement, and initially was more about like the activist side of things, and eventually my drug development work gotten more and more intertwined with longevity research or fighting aging, and got interested in the actual science of aging and the fundamental reasons why we age, and kind of went down the rabbit hole of trying to understand what are the fundamental mechanisms, what epigenetics of aging, and whether aging can be epigenetically modulated. And ultimately, I guess serendipitously at the same time became aware of partial reprogramming and the successes that partial reprogramming saw in extending lifespan of at least progeric animals. And ultimately that led me to want to start a company to develop this paradigm into therapy. Initially because nobody else was doing it, but now it's kind of all the rage these days, and it seems everybody is having
a lot of people,
partial reprogramming startup. Yeah, I think there was even a joke on Twitter. I think Nathan Chang was like joking about that maybe he should also start a partial reprogramming startup. Of course, I think Altos is kind of driving a lot of hype around this, but it's huge investment and heavyweights, entering the ring, and maybe Brian Armstrong as well with his NewLimit. So. Mm-hmm. But yeah, it's great to see that partial reprogramming is now getting a lot of attention, and hopefully it won't kind of follow in the footsteps of all previous paradigms, and will provide some meaningful life extension that will be able to translate to human life extension. But we're still pretty early in that process, but I'm very optimistic.
Yeah, 100%. Very optimistic. And I'll come to partial reprogramming next, but just so there's some context, the epigenetic theory of aging, you're obviously a huge advocate for it. And you've written about, your strong epigenetic theory of aging too. And you call, say, maybe Sinclair's informational theory the more weak theory. What is the strong epigenetic theory of aging?
I was just kind of trying to imitate physics with the strong and the weak theories, but I think the kind of my vision is that epigenetics drives a lot of aging, and I think aging is not only is it a biological adaptation, which I think very few people would argue with me about that. But it's actually, a programmed process where species are programmed a certain kind of life history, which includes a period of aging. And there's a lot of like hair splitting going on. Is it a quasi-program? Is it like the post-reproductive stage? Is that programmed, or is it like where the program ends and that post-reproductive stage is where genes don't care about it? And it could be a hours-long discussion about all the details of that. But to me, I think there's actually like all of the stages, all of the life history stages are epigenetically controlled, and I think they're pretty tightly controlled, like the onset periods. Because nobody will debate or will disagree that puberty onset or embryogenesis stages are very, very tightly controlled and have very kind of precise time points where, like embryogenesis is a very, very intricate and precise moments. Puberty onset, you got a little variation. You can like maybe a year or two variation. And then, you get a little bit of a dispersion on the actual like stages, in aging or development, post-reproductive, onset, and so on. But overall, you see a very, I think, predetermined pattern of life history in in humans and other animals, and the actual timings are they're pretty tight range. So, and obviously, to me at least, they are epigenetically implemented. So it's epigenetics, which essentially kind of reprograms you from a pre-pubertant kid to an adolescent to an adult and to a post-reproductive adult organism. And so I think if we're actually able to, 20%, if we're actually able to figure out the epigenetic controls, we might be able to slow down these transitions, or even, potentially maybe reverse them or roll them back at least a little bit, like make your epigenetic pattern of gene expression in a lot of your tissues be closer to a younger pattern of gene expression than to an older pattern of gene expression. As essentially, hopefully, epigenetically decrease your biological age. And so, yeah, basically, I think there's a lot of agreement about the role of epigenetics, but there's some disagreement whether that kind of role of epigenetics is maybe stochastic, like it just happens like because of some stochastic damage or stochastic mechanism that epigenetics has to kind of react to them, and this is why we see the epigenetic changes during aging. And to me, that's what I call kind of the weak epigenetic theory, that epigenetics is reacting to maybe some DNA damage, which is kind of one of the arguments of David Sinclair and his group that I'm addressing in that Medium post that you're quoting. But to me, it's actually, I think epigenetic changes are kind of happening by themselves. They're not a reaction to something. They're essentially an implementation of a life history pre-programmed for a given organism with set time points that just take place because of that's how the organism is set up. So epigenetics is actually driving those changes. So I think I got a pretty long-winded explanation about kind of my view about the role of epigenetics.
No, that's actually quite interesting, because when I was talking to Dr. Morgan Levine too on her epigenetic clocks, in fact, she talks about methylation patterns. She noted that they're actually not as random as you would think. They're quite predictable, and they're quite distinct in their ways, and that's what surprised her, that it's just not random. Like you can see set patterns too.
Absolutely, and yeah, to me, this is one of the arguments for the strong epigenetic theory of aging, that there is this concept of epigenetic clocks, and there is a lot of similar changes between different humans, different mice of the same age, that you actually can build a clock that will accurately tell you how old a given tissue is, or how old of an animal it's coming from, and also that it's synchronized between different tissues with very kind of different dynamics. Like there's slow dividing tissues, there's non-dividing tissues like neurons, there's fast dividing tissues like blood cells, and there's some universal methylation, CPGs, or like I guess kind of the underlying mechanisms that are the same between those tissues, that to me hint at some universal epigenetic process that is driven by aging, or at least highly correlated. And I think it's driven. Some people think it could be just reflective of something else, some other underlying process. But yeah, to me, this presence of epigenetic clocks is, I think, is an argument for the programmed epigenetic nature of aging.
Yeah, so you spoke about partial reprogramming and maybe turning back the epigenetics a little bit. You started a company, YouthBio Therapeutics, based on that, the Yamanaka factors, the OSKM, which is quite popular now, as you said, making the rounds. Can you maybe just explain what is epigenetic reprogramming or partial reprogramming?
Sure, yeah. I'm sure most viewers will probably know by now. But basically, we, as a community, we were blessed with this miracle of Yamanaka factors that while they reprogram cells back to embryonic-like state, also turns out that they are able to rejuvenate cells, physiologically rejuvenate cells in the process. And this is a miracle that I think we didn't have to be blessed with, but thankfully we are. And I think we're actually kind of stumbling on some of the life's fundamental mechanisms of rejuvenation, which are probably only available to cells after fertilization to implement this born young kind of paradigm that, every kind of old organism is able to produce a young organism only after reproduction. And it seems that, the Yamanaka factors that were discovered are at least partially responsible for this rejuvenation process that happens after fertilization, or resetting of the age that happens after fertilization and after reprogramming, which I think reprogramming kind of mimics the process of, this wholesale reset, epigenetic reset, and also physiological reset that seems to be accompanying this reset. And so the partial reprogramming paradigm arose when people started to experiment, like, can we get the benefits of this rejuvenation that we see in vitro, in a petri dish? Can we do it in vivo, in an animal? Because we are very selfish organisms. We actually want to be rejuvenated while we're adults, not for our genes don't care about our rejuvenation. They just care that our progeny is rejuvenated. But we want to kind of get that magical rejuvenation that is usually reserved just for our children. After we produce the fertilized cell, we want to try it in vivo. And in vivo, obviously we can't do full reprogramming because our cells will stop being functionally, the cell types that basically make us alive and function. And if they stop being a skin cell, if they stop being a neuron and then go back to embryonic-like state, essentially will stop functioning and die very quickly. But the partial reprogramming observation was made that what if we can actually just do a little bit of reprogramming without letting a cell lose its identity, but hopefully get the benefits of rejuvenation, like a little bit of rejuvenation that could be happening during reprogramming. And this was one of the observations that was made initially, that reprogramming and rejuvenation are gradual processes. So theoretically, we could find this kind of narrow therapeutic window, this Goldilocks zone, where the cell is still the same functional cell that is starting the reprogramming process, but it's already kind of a little bit more rejuvenated than when it started. So this is essentially the underpinnings of partial reprogramming, where we try to get the kind of the best of both worlds with rejuvenation, but without letting the cell lose its identity, without letting the organism lose the function of the tissues that are being reprogrammed.
So how does that work? How can you just turn it back a little bit and then stop the rejuvenation process and not let the cell lose its identity? Like how would it biologically work?
Right. Well, you need to activate the Yamanaka genes somehow. You could do it endogenously. I mean, you can activate endogenous genes that are already kind of present in each cell, the DNA for them. You could do like a CRISPR activation with epigenetic mechanisms. But I mean, the paradigm that we're working on is we're just delivering Yamanaka genes under an induction mechanism. Like basically, without a inductor, the genes that we deliver will not be expressed. They'll be silent, and this will prevent basically these genes reprogramming tissues at will and having us die of losing function or teratomas. So when we deliver those genes under the control of, for example, tetracycline promoter, the only time the genes are activated is when we give the animal tetracycline or its analog, like doxycycline. And so when we stop giving you doxycycline, gene expression stops, and this is what accomplishes the partial reprogramming. So basically, we only give you an inductor for, say, 2 days, and you'll only get 2 days of activity of Yamanaka genes in those tissues where we delivered the cassette with the Yamanaka factors. And yeah, so this is kind of the in a nutshell, the approach that we're using. You can also try and do like mRNA delivery, which mRNA degrades pretty quickly, and you can get kind of very short activation of the factors. But from our perspective, we actually want a gene therapy that you can deliver to the cells of interest once, and then you can do repeated rounds of reprogramming for, how many rounds. I mean, because probably this will have to be a repeated process. It's not like you can do like 1 or 2 rounds of reprogramming and you're forever young. Unfortunately, we're kind of always fighting an uphill battle with our, I think, pre-programmed process of aging and epigenetic changes that accompany it. And if we want to kind of stay in the same spot, and we have to kind of be running on this treadmill or just to stay still. I think I'm butchering the Alice in Wonderland quote. But yeah, so basically, I think if we want to keep or freeze our epigenetic age, we'll have to do repeated rounds of partial reprogramming, which kind of get it lower. And then once we stop reprogramming, our biological age will kind of revert back to the original setting. And then we'd have to do another round of reprogramming and kind of keep playing this yo-yo game until we figure out how to, epigenetically stop this process of aging from happening in the first place. But until then, I think partial reprogramming is kind of the next best thing that we could hope for if we learn to at least roll back our biological age to a lower level, and the relaxation process or the kind of snapping back process happens over a longer period of time than the interval between our reprogramming rounds.
Mm-hmm. No, that's super cool. So I'm curious, in your experiments with animals, the activation molecule you give, doxycycline, for example, to activate the OSKM genes, is the dosage or the amount, the way think about it, is that different for different animals? I'm guessing it would have to be a little more personalized, right?
Yeah, absolutely. And this would be once we move from mice. I mean, we're only working with mice or in vitro cells. And once we move to other animals, it will have to be adjusted. And I mean, there are definitely known ways to scale between species and then kind of take what you developed in mice and scale it up to humans, like the concentration of the drug that you're going to use in humans. And it's very early on. Like we actually in our hopes for the human therapy, we hope to move away from doxycycline and use something completely inert, like not a antibiotic. And but this is kind of the next step. First, we have to make sure the system works and figure out like what payload do we deliver, and then how we're going to activate the payload is going to be doxycycline inducible cassette or some other inducible cassette, some other molecule that induces it. That's already kind of the much more important concern to iron out. First, we have to make sure we know how to induce rejuvenation in a safe manner and repeatedly, and we'll scale that from mice to humans. That's a very kind of understandable drug development process. Whereas at this point, we're still in a kind of drug discovery stage. We have to figure out how to exactly get the Yamanaka genes or other reprogramming genes to do what we really want to accomplish.
Absolutely. So one of the risks you mentioned, and which comes up a lot, is the risk of cancer with epigenetic reprogramming. Why is there a risk of cancer? What is happening?
Sure. Well, you're reprogramming cells, and cells lose their identity, and if they become pluripotent, then they can then redifferentiate to any other tissue that they want, and this is how you get teratomas. Teratomas are essentially like masses of cells of different types. Well, they can form spontaneously, or they can form after reprogramming, because basically, if you induce cells to lose identity, then they're kind of out of control and can become cells of different type in a spot where they shouldn't be of, different types. So I don't know, liver cells become reprogrammed into, really nasty things. Like, anyways, you don't want to read about teratomas before bed. It's not a pleasant sight. But basically, the risk is from the cell losing its identity and kind of moving past the point of no return on the pathway to de-differentiation. I guess this would be back up the Waddington landscape. And so that's why we need partial reprogramming. We don't want the cell to stop the gene expression of the genes that are responsible for the cell maintaining its identity. Like for a fibroblast, if we reprogram the cell for too long, the gene clusters that are responsible for the cell remaining the fibroblast, they will get shut down, repressed, and the pluripotency genes will start their expression, and that cell is lost to being a fibroblast. And from then on, all bets are off what can happen to it, but nothing good. So that's where the risk of cancer comes from, and it's why we need partial reprogramming, because of course we're trying to kind of hack the magic of biology, where what it's actually doing on the level of a single cell, where it's fertilized, and of course it doesn't care about anything else. It can completely change its identity and reset its epigenetic landscape. We're trying to do that in the context of an already established organism, and we have to be very careful to not lose cell identity, because that's how you get cancer. And I mean, cancer actually could be said to be rediscovering very same mechanisms of kind of embryogenesis and self-renewal and proliferation that are usually kept under very tight control for I guess very similar reasons, that you don't really want this kind of process to be happening in an established organism, because that will lead to problems. So we're working with this very powerful mechanism that nature has invented millions, or maybe probably 500 million at least multicellular organisms, and we're trying to kind of adapt it to multicellular environment, where we want to try to reduce our biological age, but not kind of get this mechanism out of control too much, because it is very powerful, and it can lead to cells rediscovering embryogenesis in situ, in place, and that's essentially what cancer is. So we have to be very careful.
Yeah, right. You don't want to mess up the gene expression. So outside of what we've spoken in terms of partial reprogramming, the risk, or just open questions, are there any other really intriguing questions that you have that you want to see other people working on the field to accelerate?
In terms of like other paradigms of
just other open questions on epigenetic reprogramming that you have.
Well, if we're talking specifically epigenetic reprogramming, I mean, there are definitely many open questions. One of which is, for example, which other factors can accomplish what Yamanaka factors accomplish, and whether there's actually a subset of other factors in the hierarchy. Because essentially, Yamanaka factors are triggering a whole hierarchy of other transcription factors, other genes being triggered. And so can we find a cluster of a sub-hierarchy of maybe transcription factors or genes that are responsible just for the rejuvenation without the loss of cell identity, without the de-differentiation? And, for us, it's an open research project, and we're looking for factors like that. And, I'm sure other teams as well are doing the same thing. And in the context of partial reprogramming or epigenetic rejuvenation, I think that's one of the open questions, at least, for me, one of the more interesting open questions. Because, essentially, we want just rejuvenation without the risk of cancer, the risk of de-differentiation and everything else. And, I mean, theoretically, well, I don't want to say there should be there, but there could be a sub-program, like a gene cluster that is only responsible for like the fixing of various kind of molecular problems, damage accumulation, and misfolded proteins that occur in, for example, in oocytes during the lifetime that they spend in the mother for like 20, 30 years. And then once fertilization happens, this gene program could be then triggered to essentially rejuvenate the cell and kind of get it, all of the damage that it could have accumulated over those 20, 30 years to get it cleared. And we do seem to have some evidence that this is what happens after fertilization. Like there's a lot of research that looked into like misfolded proteins or other damage that accumulates in unfertilized cells that, for example, disappears after fertilization. And I think trying to actually kind of bring the two together, this kind of observation that rejuvenation happens and the observation that it probably will happen somewhere under this hierarchy of genes that get activated after fertilization, and probably with the hierarchy of Yamanaka genes that they trigger somewhere. Kind of trying to find that's to me one of the interesting kind of questions under the epigenetic rejuvenation or reprogramming umbrella.
Well, on that point, I think when you were in fact just working with one of the Yamanaka factors, which was OCT4. I'm not sure if that's still the case, or how did the experiments go with just working with that factor?
Yeah, unfortunately, it kind of didn't pan out to what we were hoping. OCT4 by itself is not enough to give us rejuvenation, or at least any meaningful rejuvenation. And this was kind of also validated by the Calico preprint, where they looked at all combinations of Yamanaka factors, and they saw that like each factor individually was not enough to meaningfully reduce biological age or methylation age of the cells, and you need at least two factors, and probably like three is I think a meaningful increase over two factors in terms of rejuvenation. And but of course the balance of loss of cell identity versus rejuvenation is another question, and the sweet spot is somewhere between 2 and 3 factors, at least according to the Calico preprint. But that was just one idea. Can we, out of the known Yamanaka factors, can we figure out one Calico was just then decided to try combinations of 2 and 3. But I think at this point we're more interested in trying different factors, just like something, as I said, lower down on the hierarchy of what do Yamanaka factors trigger downstream, other genes or other transcription factors that could be more relevant to rejuvenation and less relevant to losing cell identity. And this is what kind of we're thinking, or we're exploring right now. Hopefully, we'll have some factors to test on that front.
Amazing. Very exciting. All right. So moving on maybe a little bit to cryonics, because I know you're a big advocate. Again, you have a 101 on that for people who aren't familiar with cryonics at all or want to learn more about it.
Really did your research.
I think actually one of the most intriguing bits of the piece for me was when you give out examples of other animals who've survived in below freezing temperatures, but also examples of humans and human babies who've been rejuvenated after being exposed to extremely cold temperatures. I think it would be nice if you can maybe talk about some of those examples and explain why maybe cryonics could actually work.
Right. Basically I got kind of some historical examples of humans who lived through even stoppage of the heart and brain activity, because brain activity ceases, I think, pretty early on. I think below 32 degrees Celsius, I think you already start to lose, your brain waves are not detectable, and people were revived and were completely fine. And basically, that's one of the criticism of some people unfamiliar with cryonics give, that if your brain kind of stops your brain activity, that you won't, it's kind of lost forever. Even if you kind of try to revive the body afterwards, your personality will be lost. But of course, we got counterexamples from all those people who gotten cooled down to a much lower temperature than the brain activity, and they were revived and were completely fine. Basically, it's accidents. People who got frozen in outside in the winter, or fell under ice. Children, fell under the ice, and when they were found, they were completely, clinically dead, essentially. And then they were revived, and they were fine. And and animals, of course, some animals have adaptations, like some frogs, Yamanaka, I guess, that they can survive the winter and they're partially frozen. But of course, they can secrete their own geoprotectors, which unfortunately humans cannot. So we have to make do with some externally delivered cryoprotectants. And there has been a lot of research into that, not just for cryonics, but just for organ preservation for transplantation purposes. And unfortunately, I don't know, for some reason, cryonics just has had like a really bad rap in like normal scientific circles. So a lot of cryobiologists who had essentially a lot of expertise that could be transferable to cryonics, they almost didn't want to talk about this or endorse cryonics for the kind of fears of reprisals of their cryobiologist colleagues who completely disavowed cryonics and said it has no place in kind of in science. But sorry if I guess.
No, I'm curious. Where does the bad rep come from? Yes, is it just seen as very futuristic or something and not scientific?
I don't know. I mean, scientists have, I think, a bit of a kind of aversion to being overly optimistic, and cryonics essentially kind of is dependent on being very optimistic about the future, because it's essentially kind of outsourcing all the problem solving to future generations and saying, like, as long as we can kind of freeze ourselves or freeze our dead body in a decent enough state, then 100 years, 1,000 years from now, future generations will take care of everything. And I think a lot of scientists just are very uncomfortable with that kind of thinking, especially if you're using that as a sort of like a marketing tool or selling it to people who are probably not able to make an informed decision, informed consent about what exactly they're signing up for. Like for cryonics, if you're not familiar with biology or cryobiology, and cryonics is telling you that essentially you're going to be revived, you can't really assess the odds of that. The probability is pretty low, but of course it's better than zero. And essentially it's a huge gamble, but it's a gamble that I think it's a no-brainer that if you're gambling against zero, anything that you probably you could get above zero is already a win. But for scientists to have people kind of pay for the service of being frozen or vitrified, cryopreserved after their death, for a lot of scientists, that was a service they couldn't endorse, because they thought kind of the science is just not strong enough to make a case to sell this to the general public. But I think as long as you try to make a best effort in providing informed consent or information to a person to make an informed consent, and essentially to present it as a gamble versus zero odds of revival anything else provides, and if people are willing to pay for that chance, I don't see any kind of problem with cryonics. But cryobiologists, especially I'm not saying now, but like 10 years ago, 20 years ago, cryobiologists were a lot of cryobiologists were just not friendly to cryonics for some reason.
I see.
But
Yeah.
I think it's actually changing, and there's more and more overlap in, as I said, interest in organ preservation for transplantation, and essentially, it's the same technology that Cryonics is trying to apply to just whole body,
Whole body.
or at least just the brain, if we're talking about neuropreservation. And from a scientific standpoint, it's really one and the same. It's when this kind of becomes a service or a business that's when some scientists kind of stop endorsing the business of cryonics.
Yeah. I mean, I will say I have met more and more people who've signed up for it. I mean, mostly younger folks, but on the gamble that, if it's like better than zero, sure, it's like the only thing I have. Why not? Let me just sign up for it.
Yeah, exactly. And I think Tim Urban kind of made kind of the best
More
case.
popular too.
Yeah, for that, essentially, that if you're in a plane that's falling out of the sky and someone gives you an umbrella, or not an umbrella, but like a parachute that you're not even sure is going to work, you're still going to try it just because you have nothing to lose. And I think we have to kind of approach cryonics the same way, that it's essentially, yes, if we don't live long enough to have a meaningful rejuvenation while we're still alive, and we kind of meet with untimely death, we at least cryonics provides some sort of chance for revival or rejuvenation way down the line, which I don't think anybody can argue against science having making progress, great progress, decades or hundreds of years from now, that for at this point might seem unfathomable to us, because we just don't know how deeply our understanding of biology will develop or increase. So I think to sell ourselves short and say that all cryonics is never going to work, I think that is a mistake that basically is just displaying a kind of short-sightedness of the future. Because I think if you talk to someone 100 years ago and told them that we're going to have gene therapy and have iPhones and everything else, or satellites, they're probably not going to believe us and think we're crazy. And for us right now, saying that 100 years from now people will be able to, just based on your brain, to revive your personality and have at least like a digital copy of you, essentially your personality, just from your brain scan or your connectome or something like that. I think this is not out of the realm of possibility.
Exactly. No, I'm super optimistic, like you, that science will keep making progress, especially biology. I mean, there have been so many questions already answered, so many open ones, and people are making quite rapid progress at it. So we don't have to talk about this much, but I'm curious, like, how did you even come to think about uncovering or talking about the lab leak theory of COVID? Because you were one of the first people who brought it up, and then you came together and with a group of people to start Drastic Sciences. Like, what even got you interested in it?
Wow, that's quite a gear shift from rejuvenation into the COVID origins. That was just essentially not even a hobby. I mean, it became a hobby, but initially it was just a curiosity. And I just, like, very early on, like early 2020, when people were dismissing the lab leak hypothesis as just crazy conspiracy, as was I initially, just because I was uninformed, and I was just kind of trusting the opinion of virologists or people who were representing kind of mainstream science, and everybody said, like, well, usually you trust the mainstream science over opinions of other people. And so I was essentially kind of relying on them saying that, no, it's, lab leak is a crazy conspiracy theory, there's nothing to it. But at some point I just decided to, like, actually look a little deeper into the arguments of why it's a crazy conspiracy and why those scientists were so certain that it's not a lab leak and it's a completely natural virus. And once I, gotten a little deeper into those arguments, I realized that those arguments are very unconvincing. Essentially, they were, selling us the absence of evidence for the evidence of absence of, there's being any kind of signs that this could have been, if not engineered, just a lab leak from a laboratory that's been working on viruses just like this one for many, many years. And this was kind of that was the biggest red flag, right? The pandemic occurring in the same location where this lab is. And if you're talking about, wet markets, China, there's, thousands, hundreds, no, not hundreds, tens of thousands of wet markets in China. The odds of this occurring in the city where the lab is and those bats are not usually found are very small. So, and kind of once I started kind of digging into the facts and the explanation that I think fit those facts the best was that it's a lab leak. Of course, it's not certain. We don't know for sure either way. But to me, the more parsimonious explanation, kind of more logical explanation to explain all the observations was the lab leak. But at the time, very few people were saying this, and I just kind of decided to, as I write on many topics that interest me, I decided to write a Medium post about this and just kind of outline all of my thinking and the evidence for why I thought lab leak is a more parsimonious hypothesis. And that kind of started this whole hobby that turned into a hobby of investigating COVID origins. And Drastic came together, and I was one of the members of it, and Drastic found out a lot more relevant, novel information that wasn't available and was probably being obfuscated by some scientists out of the Wuhan Institute of Virology. And that kind of also gotten, I think, more and more, at least for me, convincing evidence that lab leak is a better explanation. Because initially I started, I was nowhere near like 90% level of certainty that I am today initially. But the longer it went, the more facts we uncovered about the behavior of, for example, the Wuhan Institute of Virology, and some things they were not saying or even hinting at something different than what was the reality. I think the more likely the possibility that it was a lab leak became for me. So
exactly.
And, this is kind of how it all came to be.
Yeah, no, I mean, it's cool that you actually investigated. I think it also partially it speaks to your mindset, right? Of just trying to crack this puzzle and not just take for granted what everyone or the mainstream is trying to tell you. And it's, similar to with your shift in view of aging as well. Like, why is aging not more controllable? What can we do about it? So that's brilliant, though. Yeah.
Well, I guess you could say there's a bit of a contrarian streak in me and many other people who do subscribe to, I guess, both, longevity, like radical life extension, and investigating COVID origins, because it definitely takes a bit of a different mindset, and I guess maybe a contrarian mindset. Hopefully not just for the sake of being a contrarian, but because you actually there's good rational foundation for thinking that, and not just want it to be different. But who knows? We all have our biases. So.
Right. Well, I think it's also more a mindset of just trying to uncover the truth. Like, what is the truth, and give me rational arguments for it, and like, what are the explanations, and having an open mind along the process. Few just like round up questions and back to longevity. I think one thing is interesting that you worked in drug discovery in Russia, Moscow for a bit before moving to Canada. Are there differences you've noted across geographies or cultures in terms of just how pharma is conducted, or maybe learnings we can take from different models?
Right. I lived in Canada before I went to Russia, and I guess moved back to Canada. In terms of, there's definitely cultural differences between countries, but in terms of drug development, it's a very kind of structured process, and it's a very international process. And even in Russia, there's a lot of expats from other countries who are working in this area. So I don't think, I mean, other than just kind of regulatory differences in kind of the Russian regulator and Russian requirements for having drugs registered, and say FDA or Health Canada or any other regulatory agency. But those differences are essentially minor, and they have been becoming smaller and smaller because actually Russia, like over the kind of decade that I spent working in Russian drug development, the Russian laws, the Russian regulator tried to get them as close to international, like FDA or European laws of drug regulation. And yeah, they're trying to kind of harmonize this process as much as possible so that essentially a drug you developed in Russia could then be registered everywhere else because essentially you're following the same procedures, the same requirements, and the data you generate it should be ideally very portable. That if Russian health agency gave you marketing authorization for the drug you developed there, you can then take this dossier and give it to FDA, and FDA should also hopefully reach the same conclusion. But there's always kind of little minor differences between just the regulatory process. But overall, it's really fundamentally it's very similar. You start out with the hypothesis, you do some in vitro stuff. If it works out, you go on to animals. And if it keeps working out, you keep your fingers crossed. You move to bigger animals, and if everything is okay, safety is okay, there's no issues, you then go into humans. Then you really hold your breath because that's where most of the drugs fail, and all of your years and millions of dollars go by the wayside. And yeah, if you're successful and the drug is safe and then it's still efficacious in humans, then you register it, and a whole new ball game begins where you actually need to sell it. And this kind of promotion of drugs is a very different beast and a very different skill set from actually like getting them to market. And it's very interesting because it's all the same industry, but very, very different skill sets are required. And of course, once you kind of know both sides of the game, you can then kind of maybe better understand how to do things early on in the drug development process, like which studies to conduct, which kind of endpoints to put in that might help you then later for promoting your drug, maybe positioning it better against competitors or some other ways you can demonstrate why your drug is better and convince doctors, prescribers, or patients why they should be taking your drug over something else. Of course, if it's a novel area where there's like a huge unmet clinical need, then it's much easier. If your drug works, it's going to kind of sell itself. But if you're in a crowded kind of space where there's a lot of things that already have been established, a new drug in that area it will take a little bit of marketing genius to be able to promote effectively.
Yeah. Well, what about advocacy for longevity in Russia? I feel like I actually come across a decent number of Russians involved in this space.
Yeah, it's actually quite large. Surprisingly, there's a vibrant longevity scene in terms of advocacy and just kind of events, even social media. There's, I think, a lot of like Russian-speaking, like over former Soviet Union people who support longevity and just participate in various events or fundraisers.
Question. What are you optimistic, excited about in the field of longevity, or just, anything else that we haven't touched upon that you want to talk about?
Well, I'm probably biased, but I'm very optimistic about partial reprogramming and just the interest that it's getting lately. I'm very optimistic that all of the money that is being poured into it will translate into meaningful discoveries and very quickly. And I guess kind of coming back full circle from what we started, I think I'm seeing a lot more of a sense of urgency in investors, first of all, and that's a good thing because they motivate all the researchers. And with the funding that is being put into this area, I think it's motivating a lot of young researchers who are uncertain which area to go into longevity in general, and fully partial reprogramming in particular, because I think like fighting for talent is maybe an unappreciated problem in biology, that a lot of people who start out, like smart people coming out of their programs, when they're looking around what to do, they tend to gravitate to the areas that have a lot of funding, like cancer, for example, or Alzheimer's. But now that there's more and more funding into fundamental mechanisms of aging, I think we might see a lot more young people, a lot more talent coming into this space. And this is all kind of how science is being driven. You need a lot of people looking at it and investigating, a lot of smart people to figure out like all of the mysteries of aging. And I think I'm very optimistic that all of this interest, and not just partial reprogramming, I think longevity in general is getting a lot more interest and a lot more investment. That all of this will translate into some meaningful breakthroughs, within the next 5 to 10 years. So I'm pretty optimistic about that.
Yeah, and I'm totally with you there. So, well, thank you so much for your time, Yuri. It's been great talking to you today.
Well, thank you.
Hey everyone, I hope you enjoyed the episode. I'm blown away by the pace of longevity research, and I want to keep bringing you great conversations with longevity scientists themselves. If you want to support all my effort and time that goes into the creation of this information to boost longevity, all resources can be found at livelongerworld.com. As you all know, aging is universal, and we can unite in this fight and soon be healthy forever. I can't wait, and see you next time.